Welcome, everyone, and thank you for joining us. For anybody on the Zoom call today that doesn't know me, I'm Chris Wenzel. I'm the current president of the LIPG, and we're really happy to have you here. We've been doing these now monthly, generally on the third Thursday of every month, and so far it's been quite a success. You know, primarily due to folks who have interesting information that they want to share, and we really appreciate that. And we hope that over the course of the year, we'll have 12 of these to fill out every month. We'll see. So take care of a little business here first, and then we'll get to the main feature of the evening. It's always important that we thank our corporate sponsors. It's, your sponsorships, keep the wheels turning for us. An organization like the LIPG is a not-for-profit. However, we do have expenses for our website and insurance and a mailbox and, a few other little items like that. But, we want to thank our diamond level sponsors, Cascade Environmental Drilling and PW Grosser. Thank you very much. And then we have a list of Topaz level sponsors: Alpha Analytical, EnviroTrack, ERM, Island Pump and Tank, Regenesis, VHB, and York Analytical. So it's important. It's important that we recognize them. And if you go to our website, you'll see a scrolling banner with the logos of each of these corporations, and you can click on them, and it's a hot link right to their website if you don't know them and you want to learn more, or you just want quick access. So I certainly always encourage people to visit the LIPG website also. There's news and announcements there, as well as job postings. When they are brought to our attention, we put them up there. Anyway, we'll keep going here. A little bit about upcoming events. As I mentioned, the Zoom meetings, we do them usually the third Thursday of every month. Our quarter two dates are the 15th of April, the 20th of May, the 17th of June. And those of you who are regulars here, you'll know that I beat the drum and say, we need speakers. So if you have something that you'd like to share, it's not hard. We don't pay much, but we do show a lot of gratitude. Couple of things we're thinking about as we move into a new phase of our lives and get vaccinated. We're thinking maybe we can again have the canoe trip and picnic. Stay tuned for that. We would probably push that to later in the summer. We're looking at some fall field trips and looking for folks to get involved and maybe lead one. I have one pet here I'm trying to get arranged, which would be a trip to the Hall of Gems and Minerals at the Museum of Natural History. But as of right now, that's still not really a possibility, and there probably would be limited interest anyway. Lastly, our board meets monthly. It's currently meeting online via Zoom like we are here, and we are an open organization. We meet the first Monday of every month, and anyone is welcome to show up, participate, find out what goes on. It's a good way if you're interested in getting involved with us. Come on out and see what goes on and meet some of us. Our next meeting is this coming 5th of April, is the first Monday in April. So if you're interested, you can send an email and get in contact with us via the contact on the website, lipgprez@gmail.com. And there's an upcoming event that most of you probably got an email blast earlier today for. There's several of us on the board for the LIPG that also sit on the board for the New York State Council of Professional Geologists, which is the organization that has been, wholly responsible for making sure that we actually achieved professional licensure in New York State. And, as they continue their mission now, what the council's really doing is working with the Department of Education to develop the continuing ed requirements under the law for professional geology, much like for professional engineers and land surveyors. There's an update which will include some folks from the board as well as our lobbyist who's spends a lot of time in Albany on April 1st. There was a typo there. And if you'd like to register and sign up, you can do it with on the New York State Council website. You can do it on the LAPG website. And we very much want to thank Alpha Analytical for sponsoring and organizing the webinar. So if you have a professional license, you might want to check this out and find out where we are. It's been a couple of years now, and this is an upcoming requirement that we'll all need to fulfill. One more event to talk about, the 28th conference on geology for the local Long Island metropolitan area that Gil Hansen from Stony Brook has been running is coming up this Saturday, April 12th. The requirement for abstracts closed on the 15th this week, and it's an online event this year, and I would encourage everyone that if you can take a little time, check it out. You know, I try and go every year. I always find some really interesting presentations, and they haven't posted the lineup yet, but I am confident there will be some interesting talks that are on the docket. And I will tell you one more thing about it is that the nice thing about it is they're short. They're a 15-minute presentation. They get, I think, 7 to 10 minutes for Q&A. So it's kind of punchy. It's like 1, 2, 3, and you can, in an hour's time, you can catch 2 or 3 presentations. Anyway, before we move on, I always like to put up a picture for folks to think about. I won't ask people where this is. I'll tell you, it's in the Adirondacks, and it is a fine example of structure controlling rivers. And what you're looking at here, on the top half of the page, you can see actually the upper Hudson River. If you like whitewater rafting, there's a whitewater rafting trip you can take out of Indian Lake off to the west of this view. And it's about 25 miles, and it's a fantastic trip. I've done it a couple of times. But what's really neat is you can see the blocky bedrock structure here, and you can see in the, whoops, I killed that. You can see, it's going to fight me. You can see that the river actually takes a couple of 90-degree turns, and that's a great example of, you know, the blocky structure and the planes of weakness influencing the erosional features. And when you're taking the raft trip, you actually, looks like the river just ends at a wall, and you go around these bends, and there's further on the trip, there's a few more, but I just think that's cool. You can see some of that structure here. So I love to share. And with that, the main event tonight, we're very lucky to have Sarah Nicholas, a scientist at Brookhaven National Lab, is going to make a presentation on chasing iron and other elements in the oceans, and she'll explain what that really is about. So what I'm going to do is I'm going to stop sharing and allow Sarah to take over, and she'll share her screen and get on with the presentation. So thank you all. Appreciate it.
Well, thank you for the introduction. I'll be talking about some of my work at sea before I came to Brookhaven. So I'm a beamline scientist at the XFM beamline at NSLS2 Brookhaven National Lab, and you may have visited my beamline if you have come for any summer Sundays. We're the ones with the dinosaur that we've measured through a piece of shale, and so that's up. So that's a sort of landmark for my beamline. During my PhD and postdoc, I spent about 23 weeks at sea doing marine chemistry, so just under 6 months at sea. Marine chemistry is the study of the chemical composition and chemical processes in the world's ocean. Some of the key processes studied are the cycling of organic and inorganic carbon and nutrients like nitrogen and phosphorus and trace elements like iron, which is what I was studying. I didn't start out in oceanography at all. My PhD work was on arsenic speciation in Western Minnesota, and I think that you have a lot of surficial geologists in this group, so you will recognize. So this is a typical day in the field before the coring truck arrives, when you kind of sit in the truck and decide whether you really want to do this or not. And then the coring truck has arrived and we've all started work. So the ground, to go out on farmers' fields in western Minnesota, you have to wait till the ground is thoroughly frozen. So January is a good time to collect these samples. And for my dissertation work, I needed to get very fresh glacial sediments. And so they let me come out when they were coring, and I had a glove box and some sealing apparatus and a tank of argon in the field. And I would take subsamples of the different glacial sediments and freeze them under argon. So this was actually the worst day ever in the field, because this is my tent where I had a glove box and it collapsed about halfway through. But then the next picture you see is me enjoying the sunset on the night before we got into Tahiti at the end of my first cruise. So it's kind of a juxtaposition. And the reason I got to go to sea was actually my work with the glove box. So I went to sea for the first time as part of the GeoTraces program, which is an international program that looks at the cycling of trace elements and isotopes in the oceans. And my dissertation advisor, Brandy Toner, had an NSF grant that was funded for her to collect samples, and she asked me to go and collect them and seal them. And so the trip that I was on, the cruise that I was on, went from Ecuador to Tahiti, which is a nice warm weather ride. So when we talk about cycling in the ocean, the big paradigm for chemical oceanography right now is the biological pump. And that's a way of describing how the cycling of carbon and carbon dioxide from the atmosphere into the oceans, into sediments, affects the global climate. And we talk a lot about the biological pump, and this is basically photosynthesis in the upper ocean. So this will be, I hope you guys can see my pointer. This is probably the photosynthesis equation as you first saw it in elementary school or in junior high, where we see carbon dioxide being converted to sugar. And if we go a little farther, and so that's happening here in the upper ocean. 90% of the biological carbon that's formed in the upper ocean never leaves the photic zone, the zone of light at the top of the ocean, and only about 0.5% of the carbon that is sequestered in the bodies of these marine plants and animals ever reaches the seafloor. So the balance of carbon for all this is it's a very small margin for these different sources and sinks of carbon. I always like to put this little cartoon up because actually phytoplankton are half of the primary productivity on Earth. We talk a lot about trees, but in fact, the surface of the ocean is so large that these tiny, tiny plants that are so ubiquitous do about half of the photosynthesis that happens on Earth. And here they are very upset about the trees. So if you get a little deeper into this photosynthetic equation, you can think of it, it's actually a redox equation. And so this is a little more complicated version of the photosynthesis equation. And what's really interesting is that it actually has to be catalyzed by a metal. And the metal for photosynthesis that almost all plants, both terrestrial and marine, use is iron. So iron is a limiting nutrient for photosynthesis. In the absence of iron, the process cannot go forward. And there's a lot in this map. So we're going to have to walk sort of slowly through it. But on the left, you see July, August, and September. So the Northern Hemisphere summer, the austral winter. And on the right, you have the Northern Hemisphere winter and the austral summer. And the row and the two figures on the right show you the distribution of chlorophyll A, which is a good proxy for photosynthesis. And the pictures you see on the bottom are nitrate concentration. And what's interesting is that nitrate is being used here as a proxy for most nutrients for plants. And what you see here is that, in general, these things line up. Where there's a lot of nutrients, there's a lot of photosynthesis. But there's a couple of places where that is not true. And one of them is over here in the central Western Pacific. So there are these places where there's an excess of nutrients, and yet the chlorophyll isn't using them. So, what's missing, I've sort of hinted at that. What's missing turns out to be iron. So this is the current paradigm. This is really most of what we talk about, it seems like, in marine chemistry. So iron is present in picomolar concentrations in the world's ocean. Almost all of the iron in the world's ocean comes from the continents. Overwhelmingly, it comes from continental dust that blows in, and it's carried in river water. Also marginal seeps at the edge of the continents, but also a small amount from hydrothermal vents and micrometeorites. And so on my first cruise, we landed in Quito, Ecuador, went to Manta, which is the port city, and then cruised down. We went down with just 4 stations along the coast of Western South America. And then when we got to the port of Callao, we started heading west. So if you look at this map, what you see are these blue dots are what we call stations. It's where we actually stopped the ship and made some measurements and collected samples. And so you can see there are 36 stations shown here on this map, going from Lima, Peru to Papeete and Tahiti. And the thing that is of the most interest here is this hydrothermal plume. So there is an underwater volcano here on the East Pacific Rise that was detected by previous cruises looking at a helium anomaly. And the main reason we were doing this cruise was to go across from upstream of the plume through the hydrothermal plume and then west, trying to follow the chemical track of the volcanic material coming up from this hydrothermal plume on the seafloor. And my job was to collect and preserve particle samples for synchrotron spectroscopy of iron speciation. So my first ship for this was the RV Thompson, and that will always be my favorite ship, even though she was at the time, she's been refurbished, but at the time, the Thompson was very shabby, but I just loved my boat. It's registered in the US. It's part of the University National Oceanographic Laboratory System. And it's 84 meters, she's 84 meters long, 16 meters wide, has science berths for 34 scientists, but we had 36 of us because the captain gave up his cabin and slept in his ready room. And like all American research vessels, it is a dry ship. My first cruise was 9 weeks. So there are a lot of different methods for collecting chemical data at sea. We use live instruments, we use archiving instruments, and we use samplers. And this is the most frequent live instrument. It's called a CTD, which is for conductivity, temperature, and depth profiler. And typically, there'll be more instruments attached, either as a freestanding one or stuck on the bottom of another instrument, like often a turbidity meter, and almost always an interferometer to measure chlorophyll A in the water, which is to look at productivity. And when you send one of these down through the water, you look at your data as it comes in. So if you can imagine looking at this, and as you lower it through the water, you see some different things change. So here green is chlorophyll. So you can see in the first 10 meters of water, there's not that much. Then it really goes up at about 25 meters, and then it really drops off. And that's about the limit of light penetrating in the water here. The red is, I think the red is temperature. I had this in notes and I can't see them now, but the red is temperature and the blue is oxygen percent saturation. So if you can imagine, you're sort of watching all the data come in, and you don't have to imagine it because it looks like this. It looks like all these people staring at a screen. And the reason we're all staring and holding our notebooks and taking notes is in a few minutes, that instrument will get to the bottom, or as far as we intend to go, and then we're all going to fight about where we want our bottles to measure, where we want to collect samples. And water sampling at depth is usually done with a couple different kinds of bottles. So water is an incompressible fluid, which means these bottles have to go down into the water open, because if you were to close this and send it down, it would be crushed. So air is a compressible fluid. And so if you can imagine, we're passing all these bottles down at depth and they're open, and then we trigger them to close. So the one on the left is a Niskin bottle, and many of you have probably seen these. These are pretty common. There's a spring in the middle, and there's a lanyard that holds the two caps apart. And when you fire the bottle, you release the lanyard here, and the lid snaps shut. And this on the right is less frequent. This is called a GoFlow bottle. And the thing that's which is very similar, but all the metal parts, all the moving parts are on the outside of the bottle. And that's handy for when you're measuring trace metals, because you would rather your sample not be in contact with metal. So here's an array of these bottles getting ready to go in, which means somebody often me, needs to go out and after all the water has been sampled, open all the bottles. So you force back all these lids and you hook them into this array of little keys on top here. And that's what controls when the bottles are fired, when you lower this rosette of bottles into the water. And here are two guys from Lamont Doherty who are water sampling from some really large Niskin bottles. And it's like milking you get in there with your samples and you have a list of what you need and you go around and you take the samples from the appropriate depths and label them. And what you can't see in this picture is that on the cruises, there's often a water czar, and he is probably sitting over here behind Marty. And his or her job is to make sure that nobody takes more water than they've been allocated by the cruise for their measurements. And on some cruises, it doesn't matter. There's plenty of water and we really don't worry about it. But on GeoTraces, this cruise, there were 36 science traveling, and there were, I think, 56 different NSF proposals that had been funded. So everything was in tight supply, and most of us were collecting samples for more than one project. The thing I worked on most, though, was in situ filtration. So this is a little different from bringing the water up. If you want to collect particles from a really large volume of water at depth, instead of bringing bottles of water up, it's easier to send a pump down and drag seawater through your filters, and then you come up with the filters. So there's a lot of parts to this, and I'm afraid it's not very elegant looking, but it does the job. So water goes in through these 4 filters at the top, and they have baffles to try to distribute particles evenly on the filters. These are filters that are about 4.5 inches in diameter, so they're really large. Here's the pump that's dragging all that water through. There's a flow meter, so you have an idea. So you need to be able to conserve how much water your particles came out of. This is a little pinger, a little Doppler pinger to keep us from dragging it on the bottom of the seafloor. And this is a stack with 36 D-cell batteries in it, and that's what runs these pumps. And so after the water comes in, is measured, and it exhausts out, it passes past these 2 manganese fiber filters, and that's for a colleague of mine on the ship who was collecting these samples for radium and measuring them with a scintillation counter on barge. So she was actively taking radium decay measurements on the entire cruise. And here are those pumps in the hangar bay. We're getting ready to put them out, and I'm afraid, they worked really well. And one of the reasons they worked well is that they just did not have any very fancy parts. So we could not trigger them from the surface of the ship. We had to figure out how deep they were going to go, how far apart they were going to be, what speed the winch speed was, and then set a timer for each pump for when we thought we would be at the right depth and the pump should start. And this is me in the background, and I'm reaching down with a broken screwdriver to short a connection in there, and that's what starts the timer. And then we also had these accompanying bottles that came with these samples for another young woman who needed a really large volume of water for thorium measurements. And because we can't control anything on this cable, like from the ship, there's no radio control, there's no way to communicate electronically with these pumps. We had a very old-fashioned set of lead travelers, which is a plastic-coated piece of lead that you stick on the cable. And so at the most we ever put down was 14 pumps. So you'd have 13 of these pieces of lead just below your pump. And here we are putting a pump in at night. And here I am putting this is the stupidest job of the whole thing, which is to put a clamp on the bottom so that all this equipment can be set on it. And here I've been promoted to moving the pump from putting the clamp on. So that was a great improvement. But we had 13 of these travelers set up there. And then when the pumps get to depth, from the surface of the ship, you put a traveler at the top of this cable and let it go. And it slides down the cable, triggers the first pump, which closes the bottle, doesn't trigger the pump's on a tiger, closes the first bottle and releases the next traveler, which goes down and knocks the next pump, the next bottle, and the next bottle, and the next bottle. And when you consider how primitive that was. It really worked remarkably well, but you really had to keep track of the timing. And it was a very strange thing for me. I had never been to sea, and then to be involved in something it was a very interesting experience, but I was shocked by how intense it was. And here we are coming up. This is a good recovery. You can see that the bottle closed when it should. This is the chief scientist of the ship, who did not actually beat anyone on the whole cruise. And here is a second set of those filters on top. This one came up with a Portuguese man-of-war on it, which was an unpleasant surprise, and that had to be cleaned off before we could take apart the filters. So here's one of these giant filters. And you can see from how yellowy green it is that this filter was collected in an area which still has a lot of chlorophyll in it, so not at a very great depth. This is a scientist named Dan Onimus. He's now a professor at the University of Georgia, but at the time he was a humble postdoc. And he is inside the clean bubble that we built in the lab of a ship. So ships do not have clean rooms. And so if you need to do trace clean sampling, when you come aboard the ship, you measure everything, you take a look around, you go to the local lumberyard, and you buy 2x4s and plastic sheeting, and you build yourself a metal-free room inside the main lab in the middle of the ship, which is what we did. I'm sorry I don't have more pictures of it. And we brought in these big HEPA filters so that we were constantly blowing clean air in from the outside. And here I am in my part of the bubble, and you can see it's a super ergonomic workstation. Very comfortable to kneel on a box all day while working in the glove box. But actually, it worked pretty well. Here's my gas for my glove box. It had to be kept out on deck. I had 75 feet of tubing between my glove box and where I ran the gas. The regulator had to be outside as well. So there's a lot of running back and forth to run the glove box. But I had some really good days. We had some very successful data collection. And here I am isolating a sample and pipetting it onto a holder. These holders were all sent to the ALS synchrotron at Berkeley. And here I am having a really bad day inside the glove box. I think I've stuck my glove in something. And my neighbor across the way took this picture of me. Here's a set of these samples that I was taking. So to do this in a glove box, you have to pass everything into the glove box. So my notebook was in the glove box, all the sampling materials in the glove box, my camera to take these pictures was in the glove box, my microscope was in the glove box. And here are some more smaller samples for a different project. And you can see here, so this is a surface sample. And as they get cleaner, we go deeper in the water. But at the microscopic level, there's actually plenty on these samples to be seen, even though these filters look quite clean. So here's the really clean group on the ship. This is the Geotraces group. And one thing you notice that's interesting is that all their GoFlow bottles are wearing little shower caps. So they get them all clean. They put on the shower caps, they take them outside and they put them on their rosette. Their rosette is made of titanium to avoid iron contamination. It is very heavy and very expensive. And they take their bottles off to do the sampling, and they bring them inside this all plastic sampling van that has been sent to the ship separately and is bolted down to the deck outside where the helicopter pad would go if a helicopter was on the ship. But we didn't have any helicopter, and there wasn't any way for a helicopter to reach us anyway, so for most of the trip. So here they are sampling. You notice they in general wear sportswear, bathing suits, and these plastic Crocs to avoid tracking stuff in from the outside. And there were some other really neat measurements going on at the same time, and they seemed to be contradictory, but everybody made it work. This is Caitlin, and she's measuring natural mercury in the water, which is very dilute in seawater, and so she has to do a lot of pre-concentrations. And then here's my roommate on the board, Gugu Rutherford, and she's using mercury voltammetry to measure selenium. So Gugu was at the lowest deck of the ship that they allowed scientists to work to minimize the amount of motion, because that would knock loose her drop of mercury. And Caitlin was in a separate sampling van out on deck, and they were not permitted to go into each other's work areas for fear of contamination. Here's Leo. He's measuring protactinium and neodymium, so he's taking these very large volumes of water to do that. And this was just weird. This is how you measure dissolved helium. So the way you find these these hydrothermal anomalies is an anomaly in the helium-3 to helium-2 ratio of dissolved helium in the water. And so Brett here is collecting a bubble-free sample of helium, which he does by flowing water through this copper tube. Sorry, I'm pointing, and I should be using my pointer. And he taps it with this funny little stickball bat. And when he's satisfied that all the large air bubbles have been removed, he clamps it with this little pneumatic vise here. And he clamps the top and the bottom, takes off the tubing, trims the copper, and puts it in a weigh box. And that is measured back at Woods Hole much later for the ratio of helium isotopes. So this was a very successful cruise, and we got a Nature paper out of it. And what we found was, surprisingly, here's the helium, and you can see that the plume, here's our cruise. Here is in the depth, the exaggeration of depth is extreme, but here is the seamount, the hydrothermal seamount that we were measuring. All these little straight lines are casts of sampling equipment, and all the little black dots are depths of the water where we made measurements. And as you look across, the big surprise was how persistent iron was in this plume. And it was puzzling and surprising because the paradigm at the time was that, yes, hydrothermal plumes have iron in them, but it goes up and it comes back down very quickly. Like they thought within a couple 100 meters, basically all the iron would have oxidized. Seawater is very rich in oxygen at depth and when it's cold. And that they thought that, okay, iron comes up, but it oxidizes, it falls down, and it really doesn't make a significant contribution to productivity of the ocean. So it was a real surprise to see that actually the plume for dissolved iron persisted out 2,000 kilometers from the vent. And it was very exciting discovery. And this is a little model of how we think that persistent happened. And basically, it seems like ligands of organic material are holding iron actually as iron too. And by being held in this kind of organic matrix, it's sort of like, it's like a snot life jacket that keeps that iron buoyant and allows it to be transported farther. So it's kind of a sticky, if you can imagine sort of a sticky carbon-rich organic goo that the iron is embedded in. So that was my first once-in-a-lifetime opportunity to go to sea. I was very excited to get to go. I did not expect to ever get to go again. And then about 7 months later, I got a second once-in-a-lifetime opportunity to go to sea. Because a young woman in my department who was actually studying oceanography and was meant to be going to sea, her mother became very ill and she couldn't go. And so I took her place on this cruise. So this was off of Hawaii. This was at Lauihi. And you can see Lauihi down here. It's the newest Hawaiian island, and it's still about 2,000 meters below the surface. So you can't really visit it yet, but it's on its way up. It's a very active volcano. And this was unusual. This is a private research ship owned by called the Falkor and owned by the Schmidt Ocean Institute, which is the Google family, Eric and Wendy Schmidt. And this trip was only for 2 weeks. The ship is much smaller. It's the same length. It's much narrower, 83 meters long, 13 meters wide. And there were 18 science berths on the ship. So in addition to the crew, they could carry as many as 18 scientists. And this is the track of our cruise from Oahu out to just off of the big island of Hawaii. And on this trip, I got an assistant, which was wonderful. His name is Mike. And here he is working hard as he did the entire 2 weeks. And we had a much smaller glove box to work with on this ship. So this is a little tiny filter set inside the glove box. Sampling on all the research vessels goes on pretty much around the clock. And here we are on the deck sampling bottles from this rosette of Niskin bottles. Another exciting way to sample, so we talked a little bit about instruments and we talked a little bit about bottle samplers. You can also collect samples using an ROV, which is like a mini submarine. And there are sediment traps and moorings, and all of these things can be used to collect particles. And on this trip off of Loihi, we had an AUV, which is an autonomous underwater vehicle. This one is called Sentry, and it's owned by Woods Hole Oceanographic Institution. So, and Sentry was like the princess on the ship. Everything that happened. So all the work that I did, everything that anyone else did, had to fit into the schedule for when Sentry was going to be on deck and when she was going to be in the water and when she was going to be recovered. And it's autonomous, which means that there was a young woman in this group of engineers who wrote a program for Sentry every day that it went out, and it would sort of follow this program and make rudimentary decisions about how deep it should go and how far out it should go and when it should return. So Sentry would go on missions of 14 to 18 hours and then be recovered. And there she is being recovered, but maybe being lowered into the water. And then, so after that second cruise, I really decided what I wanted to do was chemical oceanography. And I finished my PhD and got a postdoc in Ireland. But my first cruise with my Irish postdoc was from Chile to New Zealand. So this was my third once in a lifetime opportunity. This is what my husband always says when he has to talk about that. He said, okay, once in a lifetime is great. And the second time he's like, really? And then by the third time, he just was unable to see it as a temporary thing. So this is the track of the third cruise, going from Antofagasta in Chile to Wellington, New Zealand. And the stations are marked here in the orange arrows and the green dots. And this was on a German research vessel, and I was responsible for a lot of measurements of things I had never measured before. So it was an exciting time, and I learned a lot on this trip. So like, in addition to iron, which I was used to measuring titanium and iodide and via voltimetry, and then peroxide and superoxide via fluorescence. And then I also took and preserved samples to bring back to the National University of Ireland for measurements there. And so this is this German research vessel. It is enormous. It's huge. These tiny little people are people. And this is the ship at the dock. It was half again as big as any ship I'd ever been on. 120 meters long, 21 meters wide, had 40 science berths. It was just an amazing facility to work on. To give you an idea, this is the entirety of private space I had on my first cruise on the Thompson. This is my bunk, and I can pull a curtain if I want to be alone and have a reading light. These are some pictures my children drew. And that was it for privacy. And then I get on the Zana, and I had an entire cabin to myself. It was about 12 by 14. I had a porthole with a view. I mean, it was above, it wasn't just looking out into some equipment, it was actually looking out on the water. I had a beautiful mahogany bunk that was sort of built into the curve of the ship, and I had my own bathroom with a heated floor, and a passive-aggressive German steward came every Thursday and cleaned my room for me and judged me, but he also cleaned my room. And here I am away from the ship. Just this is unusual to be able to leave a ship in the middle of the Pacific, but I was collecting samples out ahead of the ship. So just get an idea of how enormous that looks out on the sea. So after that cruise, we returned to Ireland. I returned to Ireland, where my family had been waiting for me since September. We moved before my first cruise so that the children could start school. And this is sometimes called the real map of Ireland because it shows the range of the Irish Shelf and the Irish maritime rights area for fishing. Here are my kids getting to know some of the locals, and they had a really nice time. We lived in Ireland for 2 years. And the ship I was on in Ireland is the Celtic Explorer. It was the smallest of the ships I'd been on. It was 66 meters long, 15 meters wide, has 22 science berths. And unlike the other ships, which were really dedicated to geochemistry, the Irish research vessel is dedicated to fisheries research for the most part. But if there's room, you can come along and collect some water chemistry. So since they were doing this boarfish survey, which they do every year, and here I am with a boarfish, it was basically like mowing the lawn of the west coast of Ireland and Scotland. And the stations where we did chemistry are marked here in these spots. With the circles in the boxes are where I collected radium samples and started counting them at sea. But most of my water sampling was simple tests for nutrients, and this is a colleague of mine being delighted at a color change in a sample. But my main job that was actually related to what I was meant to be doing on the Boarfish cruise was measuring radium. And I think if you remember from the very first slide where I showed some filters sitting on top of that pump stack, that was how Virginie Saniel was measuring her radium. But because we had a more abundant water and no real way to send bottles down to such depth, I was measuring radium on the Irish ship by bringing water up and filtering it. So I would bring water up, put it in these giant water butts, and then run it through, pump it through filters and count scintillations with a scintillation counter on board for that. And so these are my enormous, so here we would call them rain barrels, and in Ireland they called them butts. And it was a source of much amusement that I had these big butts and I needed to strap down my big butts. And has anyone seen Sarah's big butts? And all this went on for 4 weeks. The Sir Mix-a-Lot song but it was very popular at the time. And so I heard a lot about that I had big butts, and I could not lie. But I collected a lot of samples, and I counted them on board, and then I continued to count them for about 18 months. And I also collected freshwater radium. Well, radium is a signal of freshwater, I should say, for the most part. So here I am on the Clare coast where fresh groundwater is coming up as submarine groundwater discharge. And so I also measured radium there. And we had some very interesting results for the radium anomalies in around the Aran Islands. And this is coming to the end of my time in Ireland. I also did some aerosol measurements off the sea at Mace Head, which led to more synchrotron measurements, which led to me being offered a Stony Brook postdoc, which led to me getting my job here at BNL. So I ran away to sea and did something completely different than what I had been trained to do. And there are lots of student opportunities. So if you have students, and there are some volunteer opportunities. So it's possible that you could also run away to sea if you would like to. So there are some volunteer opportunities to the US laboratory ships. And there's this interesting program for engineers where they feed you and house you and even pay you a salary for a year. And you get to sail all around the world to interesting locations. And almost all of those interns decide they want to be marine technicians and take jobs on research vessels. So that's typically for engineers. And there's also student opportunities through the Schmidt Ocean Institute. And interestingly, they also carry an artist on most cruises. And so you can find, there'll be a whole group of chemists or biologists, and there'll be one person sculpting or painting or doing really interesting photography. And all of this has changed a little in the last year because crews are smaller. I have a friend who is in the process of quarantining down in San Diego for 2 weeks before she goes to sea. So the pandemic has really changed how life is at sea too, because we were in very close quarters, flying in from all over the world, having a couple days before we took off and were on a ship together, hundreds of thousands of miles from land. So naturally, COVID has really changed how these cruises are done. But my hope is that we will get back to normal and people will get back to sea. So this is just my winding up slides. Here's a sunset on Tahiti. Here's a sunrise on Tahiti. Here is my favorite sign in all of Ireland. Does anyone have any questions? So that is the end of my talk. I should say. Okay, I don't know how to, there we go.
Where in Ireland did you live?
Galway.
Oh, nice.
Yeah, it was beautiful. We really loved it. My younger child has been accepted at the university there where I was a postdoc. And so my hope is she'll be able to go back there in August and start.
So now when you were tracking iron across that plume.
Yeah.
Are you able then, does that allow you to look for other plumes based on iron concentrations? Is that the purpose for
Well,
what you're doing?
so we were taking water samples as we went along, and there's some measurements that you can make by you pre-concentrate a sample and then on these little resin beads, and then you wash it off in acid. So you can get it's not a live measurement, but you can have a measurement within a couple of hours. And so we were sort of feeling our way, trying to figure out where the middle of the plume was and trying to stay in it. But we didn't find a second plume on that. So they're usually found by the helium anomaly, which is where people notice that there's way more helium-3 than there should be in the gas.
So the helium is actually a tracer gas that's coming out of the hydrothermal vent.
Yes, it is enriched in helium-3 relative to helium you find in the atmosphere.
Interesting.
So, because I think is helium something, it's quite a bit less than 1% of the dissolved gas in the atmosphere, and that's also true for dissolved gas in seawater. But there's this isotopic enrichment of helium that indicates hydrothermal activity because it's mantle gas coming out. So, and it doesn't seem like a very efficient way to track a hydrothermal vent, to, take samples along a long transect, go back and measure them and see if anything's funny, and then take another track and go back and see if you can find it again. I mean, it takes a long time. I think that the vent that we went to was suspected in the 1970s and confirmed in like 2003. And then our cruise was 2012, 2013.
Huh. So all this mapping, actually, you can use it in addition to just looking at the chemistry, which I think it's amazing that it's persistent and measurable over such a great distance. You would think there would be more mixing in the ocean to dilute it, but it's also helping you map deep sea ocean currents.
Yeah, although those are, yes. And that although a lot of the ocean current mapping is done with, I didn't talk about this kind of instrument. There are instruments that you just sort of turn loose as floating buoys, and they get into different currents, and they're followed with GPS, and then they get picked up. So that's one way that people track currents. And then deeper currents, there's a set of these buoys that actually dives and comes up. And it has a cycle of diving and coming up that's about every 2 weeks. So some poor graduate student waits to see a ping that indicates that it's come up, and then it goes down again. And that's how they figure out where they are. And those get into currents and can travel really long distances deep below the surface.
That's pretty neat.
And people use gliders too, which is it looks like a little missile, but that's like a little robotic sensor, and it mostly is just measuring pressure differences on one side of it from the other, and that's how it tells where the current is.
So what method did you actually use to determine the concentrations of iron in the water?
So at sea, we did this pre-concentration Ferrozene method, and that was to sort of figure out where we were in relation to the plume. And then for the dissolved iron, that was carried back in acid clean bottles to Rutgers University and measured on an ICP. And for the particles, we had those filters, and we know how much water went through them. And then we cut the filter with careful geometry. You cut the filters into sections of known area, and some of those filters were dissolved to figure out how much iron, the concentration of iron that was held in particles. And I measured the speciation of the iron with the synchrotron at Berkeley by taking the microprobe to each little particle. So I didn't really measure the concentration. I tried to figure out what kind it was, but the concentration was measured by wet chemistry.
Gotcha. Thank you.
Sure.
Anyone else?
Did you
And I
figure
thought it
out
was
that there's-
fascinating with being out to sea at my graduate school in University of Buffalo was a lot of professors that were in Woods Hole. And I heard
Oh,
all kinds of stories about the sea. And I always thought, boy, that would have been an interesting life choice if I had gone in that direction.
well, they still take volunteers on these ships. So you could still have that opportunity. You could, and especially since you can actually, you would know something about it, which is a convenience compared to undergraduates sometimes, who don't really yet have a great idea of how to make measurements and what standards are and things like that.
Yeah, that must have been a great adventure.
It really changed my life. And the problem was I came back from the first cruise and my family was very glad to see me because I'd been gone 9 weeks, including over Christmas. And we sat down at dinner and I said, I think I want to do this instead. And my younger child, who was 8 at the time, put her head down and just sobbed. And I thought, well, I have got to work on my sensitivity, because it just didn't occur to me that everyone else wasn't having as good a time as I was when I was at sea. But that was the longest cruise I ever went on. Communication is really bad in the Central Pacific. You can have an email once a day and they spool it on and spool it off. And that's it, really.
Yeah, that had to be a hardship.
It was, yeah, I was having a good time, but I think my family, when you're the person who goes away, you're so busy doing the thing. I did miss them, but not the way they missed me. And so it was pretty insensitive of me to come home and say, I thought we should just do this forever.
Now, what do you do at Brookhaven National Lab?
I'm a beamline scientist. So I do synchrotron measurements like the ones I did after the cruise. But I'm a staff scientist, so people write proposals to come here and measure their samples on my instrument, and I show them how to prepare their samples, and we get them loaded together, and I show them how to make their measurements, and I typically stay with them, completely for the first day, and then I'm sort of in my office or nearby doing something, for the rest of the time they're here. So they usually come and make measurements for 3 or 4 days at a time. But because of COVID, we make all their measurements for them right now, and so I'm sort of being like a graduate student again.
Sarah, I have a question on that SMIT research opportunities for the students. Do you know anything about that? Can you speak on that?
I can try to get back to I think I can go back to the slide and there's a link.
Yeah, I tried to jot it down.
Oh yeah, well we can just go back and I think I can go back. Let's see if we can.
Yep, that's it.
Oh yeah, I see it's sitting right onto https schmidtocean.org.
Yeah, I mean, have you had students that have gone through that program or not?
No, I was on because we had a NSF grant to go, or maybe I don't know how that worked actually. I was on because my office mate wasn't able to go on that cruise, but I think that Brian Glazer, who's a professor at Hawaii, had written the proposal for this cruise. But so professors write the proposals and they take their students, but the ships often need more people than than faculty are able to send. And so it's an opportunity there used to be just a list. You would just put your name on the list of volunteer opportunities. But I've never actually done it that way. I've always gone on because somebody with a grant needed someone to make a specific measurement and they wanted me to do it.
Is it open to undergrads or just grad students? Or?
I think that this Schmidt one is open to undergrads.
Okay.
And I think the UNOLS one is open to undergrads also. And in fact, my understanding was it's also open to fairly recent grads. So people who you can go do it. And there used to be just a list of volunteer opportunities and the extra berths would come up, 3 or 4 weeks before the cruise was going to sail. And they would call names basically off that list. And So it could be doing, typically the volunteers do fairly simple measurements, like they might help with the salinity measurements, which go on all the time. Logging temperature, it might just be, basically helping to pull things up out of the water, that kind of thing. But, the food is really good. On all the ships I've been on, the food is excellent. The people are nice. It's really interesting to be involved in a 24/7 endeavor with a lot of other people.
Well, I teach at a community college, and I teach actually oceanography is one of the courses I teach.
Oh,
And and we have students who are interested in oceanography, and we send them off to four-year schools to do oceanography. But there was never really didn't know a lot about any research opportunities or programs they could get involved in at the undergrad level that we could probably introduce them to. So
well, I think the UNOLS one would be a good one too, which is this one, because this is Schmidt is just one boat, and but UNOLS has, I think, more than that. The Neil Armstrong, the Sally Ride, the Thompson, the Roger Revelle. Those are the really big interocean ships that are big enough to carry the large submarine, the Jason submarine. And then there's a bunch of coastal and mid-range ships. So there's a lot of research, and these are all organized by the University National Oceanographic Laboratory System. And these ships are actually all on long leases from the Navy.
Okay.
But they're not as cushy, certainly, but they go out a lot, and they have a lot of different universities will have research projects they're running on them.
Okay, thank you. I'll look at the website when I get it.
This year is not a good year for it.
Yeah, this year is not a good year for anything. So.
No, but my hope is that things will improve again.
Yes. Hopefully, by this time next year, things should hopefully be looking up.
Okay.
So
I'm going
but thank you. I enjoyed your talk immensely.
Good. Where do you teach?
Onondaga Community College in Syracuse.
Oh, wow. Okay.
Yeah.
I haven't even been that far north, I don't think, yet.
We're
in New York. Maybe I have.
If you've ever been up to Syracuse University, we're in that same area.
Mm-hmm.
So
okay.
We feed Syracuse University lots of students in ESF. So,
oh yes.
So, but yes, I liked your talk. I found it very interesting.
Oh, thanks.
And I'm teaching a section of oceanography this semester, and next week I'm starting geochemistry or ocean chemistry. So this was a good little refresher for me too.
Oh, I'm glad.
So, but thank you. I enjoyed it.
Oh, well, thank you for coming.
Anyone else?
Who is 0212?
Don't know. Anonymous.
Oh, okay. Well, that's fine, but they're right. That isn't, the weight is not called a traveler. It's called a messenger. 0212 is right about that. But I'm really curious because they say they remember hanging Van Doren bottles, and so I'm wondering who that is. But I guess I'll just have to wonder.
Strong silent type.
I guess so.
Okay, well, Sarah, thank you so much. That was a really interesting presentation, and it's really nice of you to share your experiences with us. I think that was really cool and insightful. And we sincerely appreciate you making the effort.